Hi, everyone. Okay, so this is Professor Youngjin Park, who's a computational biologist and an assistant professor in the Department of Pathology and Laboratory Medicine at the Department of Statistics at the University of British Columbia. Professor Park, I'm just going to allow you to introduce your own research. Can you tell us what are your current research interests in terms of genetics?
Hi, yes, my current research in genetics now has to do with a lot of complex traits. So I'm trying to understand what's the causal mechanism of the complex traits, such as a lot of neuropsychiatric disorders. And sometimes it can be your lifestyle, sometimes it can be your cancer predisposition, cancer risk, and so on. So my goal is to try to figure out what is the molecular underpinning of those complex disorders using genetics and molecular biology and computational biology as well.
Okay, and I'm a bit more biased towards neurodegenerative disease. Can you tell me what kind of diseases maybe that you study?
So during my postdoc with the Manolis Kelly's lab, I mostly worked on Alzheimer's disease. One of the things I noticed was Alzheimer's disease is not a single disease. It's basically a complex disorder. It can have multiple types of phenotypes. There are multiple regions. You may end up being, dementia. So my first goal was trying to figure out how do you classify different subtypes of Alzheimer's disease, whether it's more neurodegenerative or whether it's more cognitive decline side. So that sort of thing, first I worked on it. And then later I moved on trying to understand, what is the causal gene? And if the causal gene was there, and what's the relevant cell types, then you may be able to understand the molecular mechanisms by knowing causal gene and the causal mechanism. What is the regulatory element that drives the changes in the causal genes, disease risk, that's all.
Yeah, I'm gonna have to agree with you on that. A lot of people believe nowadays that Alzheimer's can be caused by multiple things. Right now, I'm also interested in a combination maybe of different genes that could be contributing to an Alzheimer's phenotype. And while we're on this topic, can I ask, to what extent do you think that genetics and epigenetics are responsible for neurodegenerative disease?
So first of all, we have, I was fortunate enough to work with the Manolis Kelly lab. They have profiled all these epigenetic roadmaps. And at the same time, we also had other in-house case control data sets, both histone modification and DNA methylation, and also transcriptome as well. So those things actually really point out that, those neurodegenerative disorder can be a lot of variability can be explained by epigenetic enrichment. At the same time, one thing that we were able to know is surprisingly, it's not really neuronal in many cases. So a substantial fraction of variability was explained by immune relevant tissues and cell types. So in that case, it's good to have epigenetic signatures because that can actually nail down what's the disease etiology. So most people have not really considered it before the epigenetic analysis.
Yeah, that's interesting. Now, when you say it's not neuronal, you're referring to glial cells like maybe microglia or astrocytes, right?
Right. So another thing is when you look at the phenotypic landscape of Alzheimer's disease, for example, there's other pathological variables as well, such as your aging. So some people may undergo healthy aging. Some people may have, some your brain may be unusually older than your actual biological age or a chronological age. Then in that case, abnormally accelerating aging is another component that may, expose your risk of Alzheimer's disease more frequently than the other case. And I guess in my case, another thing potentially interesting to look at could be there is a different ethnic groups. You could have a different disease etiology as well. So some people may undergo, it could be environmental, it could be genetic, but some group of individuals, ethnic groups may undergo more severe or more stressful life event. Then that will put you in the boundary of in the disease risk spectrum.
Yeah, there's so many things to consider, right? Like it's so complex, so interdisciplinary. Sometimes it seems like you're not really sure what to focus on to come to the root of the disease. So I guess a question for you would be, what are some of the most pressing questions in your opinion to answer about aging and neurodegenerative disease? Like say if you had to choose 3 questions that you're curious about, what would they be?
So I think that it's mostly, it's not just neurodegenerative disorders, all the human common disease or complex disease. It's really challenging because we cannot make interventions. So we cannot make people to assign some Alzheimer's disease, for example. It's not a treatment assignment. So we mostly have to deal with all these observational data. So that it's an accumulation of all your exposure to risk factors. So sometimes your genetic risk factor may be interacting with all these environmental risk factors, but sometimes it may be beneficial. It can be protective against your environmental factors as well. So those things are really hard to disentangle in some sense. So that's actually the challenge. And then in that sense, genetics is probably unique data set. So because it's sort of clearly teaching us what's the causal directions. There's no way you can your disease can impact on the common your genetic variants. So that's actually unique. But another problem is one downside of the genetic analysis. Its effect size is usually very small compared to other environmental signals.
Yeah, I think it's important as researchers in this particular disease to kind of be open, and I think that's where genetic analysis will be very useful because it's more global in targeting the cause of the disease. And on that note, I guess we'll shift to the computational side of things. I understand that you are an expert in machine learning. What types of data do you think that artificial intelligence and machine learning or deep learning are best suited for? And what types of data do you think they're not suitable for?
So, machine learning I view it as statistics. So basically you cannot create something new out of nothing. So that means your data experimental design is very important, but this is an observational study. So that means it's very important to obtain balanced data. So one of the key component of success behind the success of GWAS was because people were able to form an international consortium that you could have a fairly well balanced case control studies. But as we move toward biobank type of data analysis, you don't have control for forming consortium and so on. It's just observation of prospective cohort. So in that sense, it's really important to how to weight each individual observation differently. For example, some people it's probably more informative than the other people. So in terms of disease classification. So that's something you may consider as in the fairness perspective. So in the machine learning, you want to guide your training algorithm to more fairly classify what's disease versus non-disease and so on. So that sort of thing is a very important question. So that machine learning algorithm needs to address.
Yeah, those are really good points. And it's also really interesting that you bring up that this type of work is really dependent on the collaboration of people not within just institutions, but also worldwide, right? So, and I know that you also do some work in polygenic risk scores. Is that correct?
Yeah.
So how do you see polygenic risk scores applied within the domain of precision medicine? Or what role do you see polygenic risk scores having in precision medicine in the future?
Yeah, I think polygenic scores it's still too early to be practical. Last year there was a paper showing that the polygenic score model of your height you train on, for example, UK Biobank, which mostly European ancestry, which is actually useless if you apply the same model to Japanese biobank, for example.
Oh, that's interesting.
So, yeah, it makes sense. I mean, height is one of the trait that's so easily predictable by genetics. It's heritability can be explained up to like 80%. Your heritability can be explained by common variants, but still, depending on how you calculate. But still, it's suddenly you're changing one data set to the other data set, different ethnic groups, for example. Then it's really hard to be more practical. So for a precision medicine, I think it's still too early to use polygenic scores. But if you, instead of just training your polygenic score as it is, like a black box model, but if you use polygenic scores in a more, biologically guided way, like say that you construct the polygenic scores, only combining those, enhancer locations that are, mostly relevant to neurodegenerative disorders, for example, then it may be universal across different populations. So that it's more, it can be more robust, like in terms of, feature selection in machine learning perspective. So in that case, it can be useful, but it's a polygenic risk score, which means it's your risk prediction, but it doesn't necessarily mean that you're gonna get the disease. You have a, you can be more exposed, but it's not really a decisive way. So I don't think it's really a tool for precision medicine. But as we predict disease risk more accurately, so then maybe that can guide you to more chronic conditions. But like some people may sometimes could be born with some higher risk of some particular types of cancer, for example, or some diabetes. In that case, you can use it for like a more public health perspective. So preventative medicine rather than precision medicine, I would think is more like a more individualized.
Yeah,
it's more like guided.
yeah, those are good points. And I think from the outside looking in, I think it's also easy to take for granted how much work is involved to take something from bench to bedside. For example, in Alzheimer's disease, we also have the biomarker panels are still using largely three biomarkers that we've known for so long now, which is just amyloid beta, total tau, and phosphorylated tau. So regardless of all the progress that we've made in identifying more biomarkers, we still have a lot of work translating it into something that can be used in the clinic.
Right.
I agree with you.
But I guess it may converge at some key pathways, which we have to still investigate.
Mm-hmm.
But then in that case, your polygenic score is more interpretable. So.
And on the topic of GWAS, so GWAS, for readers who aren't familiar, that's an important analysis for identifying disease-causing genes. So from your experience, what do you think are the current limitations of GWAS? And what can we do as researchers, as a collective, to improve it?
I think because it's more like a design choice, I guess. But it'll be much more beneficial if you engineer your phenotypes more precisely rather than case control. So since we are entering biobank era, so which means we have all the other biometric measurements as well. So then you'll be able to engineer phenotypes more more intelligent way. So in that case, your phenotype can be really precise, pointing just one thing. Then you'll be able to come up with the case control more precisely. But there is the downside is if you try to do that, then you're going to end up being very narrow range of people, which it may not be so broad impact to as a public health value. So it's a trade-off. That's something you have to balance. So and then also your statistical power would decrease. So then you may have to balance. There's some subgroups of individuals. Then this is a particular phenotype that's really specialized for those individuals. Then your GWAS can be definitely more boosted. And then another issue, the GWAS is linking between genetic variant, which is the top of the causal mechanism, and then down all the way down phenotypes. But there's other intermediate phenotypes as well. Like in the Alzheimer's disease, we know that the APOE and then all kinds of amyloid beta or everything, along the causal trail, we know how to measure the biochemical or pathological variables. So those things are very valuable. So, but other than neurodegenerative Alzheimer's disease disorders, there's still a lot of pathological variable or lab test variables you can actually get an access, and then that will actually help your GWAS more richer and more going beyond the linear regressions, linear models, so you can go beyond that. So we need to work more on the precise phenotyping. So
that's interesting. Do you think that at the experimental design level, do computational scientists typically work side by side with these clinicians to design these biobanks?
So, yes, I think for EHR, it's a still hot area, I guess. For electronic health record, there's a huge effort from the database people and the computer science people. Sometimes doctors basically just write down or just record all this prognosis or diagnosis, and then it's more like a natural language processing. You need some CS people will jump in and then sort of parse out all this text data, and then sort of come up with the disease labeling, so in an automatic fashion. So those things are something, it's still ongoing effort. I know the Mayo Clinic and the MGH, a lot of huge, general hospitals, they put a lot of money and efforts to make it automatically, So I think that's something, a lot of the, and also the clinicians can immediately give a feedback. So whether this classification or things are, making sense or not. So basically we want to go beyond the ICD-10 code, and then we can get much richer, description of the patients. So
yeah, that makes sense. And when you work with databases, what kind of problems do you encounter? Like, what do you wish that you could have fixed retroactively?
I don't really have a hands-on, first-hand experience for this, because I mostly just help people, just chatting with people who are really working on this direction. But one thing I noticed was, all these privacy issues, it's really difficult to deal with. I totally understand that it takes time, but sometimes it takes more time than we thought. And then also the data sharing part, because it's a very sensitive data, you don't want to share with the public. So, and then all the computation has to take place within their server or some cloud space. So I think that's kind of a more like a communication issue, more like a data transfer issue. That's really difficult. So I know that people have been developing all this complicated natural language processing, all this deep learning machine learning, frameworks. I mean, I'm pretty sure that we can get more benefit, but you have to really guarantee that it has to take place in a secure manner. So yeah.
And I guess this is just going to be my last question. When it comes to Alzheimer's disease, what kind of information do you think that people should enrich databases with that would help with your analysis?
So as I did, all the brain disorder, yeah. I mean, it'll be beneficial if you have a wide range of cohorts. So, but we only have mostly we have a very old. It's more like donation based, right? So the range of age and then the ethnic groups, cohort is usually very limited. So that sort of things, we can. I don't know, but there's no other way. I mean, but because really, they volunteer to donate their samples, but it'll be better. It'll be more beneficial. And then I know that in genetics, one of the approach we can take is a proxy data, which is basically if you have a parent family history, your maternal or paternal disease diagnosis of Alzheimer's disease, that will be hugely beneficial for the next analysis, because we will know that what is the actual penetrance of the some disorders, and then then we can also trace back and build some sort of tree structures, and then figure out where and when does it really happen. And then also another thing is that sort of things. But another thing is since we have we already sort of realized that the Alzheimer's disease is also largely due to microglia, or some other glial cells. So it's very interesting to compare or contrast with other complex disorders that are has to do with the innate immunity or other immune disorders. It's very easy. It'll be very useful to contrast against them. And also I know the MIT the huge effort is going on, but it's we have to do more single cell analysis for RNA sequencing or single cell ataxia or all kinds of single cell studies. Sometimes you could have a single cell protopsy, which is basically you can immediately test what's the causal genes or causal regulatory element by perturbing particular genes or regulatory elements, and then look at their cell level downstream effects. So those things are very valuable resources, and that will definitely help understand. And then but I also think that we still need to keep all this animal model things as well, because that's the only way we can really make interventions or knockdown and so on. Then you can actually really test your hypothesis in a more reasonable way. So yeah.
Do you find significant differences between animal models and human models?
I don't know. So I'm not a really hardcore neurobiologist. So I guess they kept mostly recapitulate the common characteristics, and then depending on different types of animal models. But at least the animal models that Li-Hui side lab maintains looks like it's working really well, and it also show not only the Alzheimer's disease but also like generally learning and memory in general. So it sort of highlight what's the share mechanism. Presumably, yes, we would want to work on more. I don't know, maybe primates and so on, but I don't know. So, but they're very, very expensive. So
They're very expensive, yeah.
and it takes a lot of time. So yeah. But animal model is good, and also I know the folks in the Novartis maybe, so they are working on iPS derived different cells. So those are good proxy for looking at your cellular phenotypes as well. So
Okay, well, thank you. That's all the questions I have for today.
Yep.
I think viewers will learn a lot about GWAS and polygenic risk scores, and just the field of neurodegenerative disease and genetics overall. So thank you so much for agreeing to be interviewed today. That's it.
So, yeah.
Any last words you want to impart on anybody viewing this interview?
Not really. So
Okay.
yeah. So I think one thing I would like to mention is neurodegenerative disorder. I think it's useful to look at the other things, neurodevelopmental disorders. So it's the other spectrum, I would say. So I think also neuropsychiatry is another component. So what I also realized was there's some shared mechanisms. Also, there's some unique aspects for each of these different categories. So it's currently mostly just based on the experience based, right? It's just medical doctors examine patients and then they look at it and classify. Maybe they have a different behavior difference and so on. But as we collect more data in a single cell level, so I think it's not only just phenotypically characterized, but also their distinction is more. It looks like it's more molecular and more genomic, or transcriptomic or epigenomic, and so on.
So you think that we can learn things from neurodevelopmental diseases that can help us with neurodegenerative diseases and vice versa?
Maybe, yeah. Maybe you're making your clocks in the opposite direction or something like that.
That makes
I don't know.
sense.
That's my random thought. So.
Okay. Well, thank you so much. I'm going to end
Thank you.